The bone marrow niche houses many stem cell populations, such as hematopoietic stem cells and also mesenchymal stem cells that go and help during blood production, immune function, and the maintenance of bone. Now, we can go see that throughout time and aging, they're conserved. They constantly cycle in addition to quiescence. However, they are susceptible to DNA damage, inflammation, or niche drift such as myeloid biases that can develop into AML, acute myeloid leukemia, the overproduction of myeloid cells. Now, let's talk about RUNX1, which is an essential transcriptional factor. Hey, come back here. You said you're hungry? Knuckle sandwich! Hema stem cells are imprinted by embryonic stem cells, which they differentiate from. Once these cells are formed, epigenetic markers are found on the genome that dictate cell function. Now, how are these maintained? Polycomb repressive complex 1 and 2 repress transcriptional activity and safeguard epigenetic markers across cell life by compressing nucleotides. A recent study at the ECAN School of Medicine suggests that the PRC subunits EZH1 and EZH2 are partially responsible for hematopoietic aging. DNA methyltransferases 1 and 3 are able to upkeep the cell cycle by maintaining epigenetic integrity through de novo methylation. In concert is TET1, which aids with demethylation. What happens when these epigenetic mechanisms are no longer sufficient to keep our cells going? The carousel stops spinning, and the cell either transforms malignantly or loses the ability of stemness. Recently, epigenetic reprogramming allowed for AML to normalize the long human pluripotent stem cells, highlighting the value of epigenetics in both cancer and regenerative therapy.